Acetylcholine, or AC, is synthesized locally in cholinergic nerve endings. The process begins with the active uptake of choline by the axonal membrane through a sodium-choline cotransporter. This choline is then acetylated with the help of ATP and coenzyme A by the enzyme choline acetyltransferase, resulting in the formation of acetylcholine. Hemicholinium, or HC3, can block this uptake, depleting ACH levels. Most of the synthesized ACH is stored in ionic solution within small synaptic vesicles, although some free ACH is present in the cytoplasm of cholinergic terminals. The active transport of ACE into these vesicles is facilitated by another carrier, which can be blocked by visamicol. The release of acetylcholine from nerve terminals occurs in small quanta, with the amount contained in individual vesicles being extruded by exocytosis. In response to a nerve action potential, the synchronous release of multiple quanta triggers post-junctional events. Two toxins interfere with cholinergic transmission by affecting this release. Botulinum toxin inhibits release, while black widow spider toxin induces massive release and depletion. Botulinum toxin A and B, produced by Clostridium botulinum, cause long-lasting loss of cholinergic transmission by interacting with axonal proteins involved in the exocytotic release of HEH. Localized injections of botulinum toxin A, known as Botox, can treat various spastic and neurological conditions due to overactivity of cholinergic nerves. Immediately after release, acetylcholine is hydrolyzed by the enzyme cholinesterase, and choline is recycled. There are two types of cholinesterase enzymes in the body: acetylcholinesterase, or HE, and butyrylcholinesterase, or BUCHE. ACE, also known as true cholinesterase, is strategically located at all cholinergic sites and serves to inactivate H instantaneously. In contrast, BUCHE, or pseudocholinesterase, is present in plasma and other locations and likely serves to metabolize ingested esters. The differences between these two types of cholinesterase are significant, with HE being more sensitive to physostigmine and BUCHE more sensitive to organophosphates. There are two classes of receptors for acetylcholine: muscarinic and nicotinic. Muscarinic receptors are G-protein coupled receptors, while nicotinic receptors are ligand-gated cation channels. Muscarinic receptors are selectively stimulated by muscarine and blocked by atropine. They are located primarily on autonomic effector cells in the heart, blood vessels, eyes, smooth muscles, glands of the gastrointestinal, respiratory, and urinary tracts, sweat glands, and in the central nervous system. Nicotinic receptors, on the other hand, are found in autonomic ganglia, the adrenal medulla, and skeletal muscles. These receptors play crucial roles in various physiological processes, including muscle contraction and neurotransmitter release. Muscarinic receptors are further divided into 5 subtypes: M1, M2, M3, M4, and M5. The first 3 are the major subtypes present on effector cells and projunctional nerve endings, expressed both in peripheral organs and the central nervous system. M1 receptors are primarily neuronal, located on ganglion cells and central neurons, and play a significant role in mediating gastric secretion, relaxation of the lower esophageal sphincter, and functions related to learning, memory, and motor activities. M2 receptors are predominantly found in the heart, mediating vagal bradycardia, and on cholinergic nerve endings as autoreceptors. M3 receptors are located on visceral smooth muscles, causing contraction, and on exocrine glands, inducing secretion. They also play a role in the release of nitric oxide from vascular endothelium, leading to vasodilation. M4 and M5 receptors are mainly found on nerve endings in specific brain areas, regulating the release of other neurotransmitters.